Uh, I will wait for 1 minute, then I will start with the lecture. Okay, we will wait for 1 minute and I will start the lecture. આજે ઓડિયો બરાબર છે? Okay. Uh, I'm going to start with the lecture. Okay. Good morning, all students. Today I'm going to discuss about the specialized type of bacterial metabolisms, and which are basically chemoautotrophs, as well as some of them are chemolithotrophs also. Okay, so this includes which are the part of your syllabus are chemoautotrophs, the lactic acid bacteria, the enteric group and the related bacteria, and the archaea bacteria. In order to study all these groups, okay, you should have some basic knowledge regarding the electron transport chain. Okay, you should have basic knowledge regarding the electron transport chain, glycolysis, okay, TCA cycle, ED pathway, etc. Okay, you should have some basic knowledge as well as the photosynthesis. Okay, because these specialized groups have something more than these pathways. Okay, so starting with the chemoautotrophs. Starting with the chemoautotrophs. Okay, the groups which are included in the chemoautotrophs are nitrifying bacteria, sulfur oxidizers, iron bacteria, and hydrogen bacteria. And now I am going to start with the nitrifying bacteria. As well as before starting, I will give you general overview of all the chemoautotrophs. Okay, nitrifying bacteria. There are two types. One of them is the ammonia oxidizers, and the second ones are the nitrite oxidizers. And for the case of ammonia oxidizers, ammonium ion is a oxidizable substrate, and the product will be nitrite, and the terminal electron acceptor will be oxygen. Okay, terminal electron acceptor will be oxygen. Now moving forward with the nitrite oxidizers, the oxidizable substrate will be nitrite and the oxidized product will be nitrate. Again, the terminal electron acceptor will be oxygen. So these both group are basically aerobic bacteria. Okay, And moving forward with the sulfur oxidizers, they use hydrogen sulfide, even elemental sulfur as well as sulfides, and the oxidized product will be nitrite, and the terminal electron acceptor varies from oxygen to even nitrates. Now moving forward with the iron bacteria, okay, where Fe2 plus will be oxidizable substrate and oxidized product will be Fe3 plus and the terminal electron acceptor will be oxygen. Now moving forward with the hydrogen bacteria, the oxidizable substrate will be hydrogen and the oxidized product will be water, and again similar to that of sulfur oxidizers, they may use oxygen or nitrite as a terminal electron acceptor. Next group, the carboxylobacteria, which is not included in your syllabus. Okay, so oxidizable substrate will be carbon dioxide and oxidized product will be, sorry, not dioxide, carbon monoxide, and it will be converted into carbon dioxide and the terminal electron acceptor will be oxygen. Okay, and there are several groups of soil or aquatic bacteria which are able to use inorganic compounds or ions. Okay, and they may use ammonium nitrite, sulfide, thiosulfate, sulfide, or a ferrous ion. Okay, some can also use elemental sulfur, hydrogen, or carbon monoxide as electron or hydrogen donors, and gaining energy and reducing equivalents for the synthetic process which which are done by the oxidation. And the energy usually in these cases can be obtained by the respiration with the oxygen as a final terminal electron acceptor. Only a few specialists in this group can grow with nitrate or nitrous oxide as hydrogen acceptors. That is basically a type of anaerobic respiration, and this mode of life with inorganic hydrogen donors is called chemolithotrophy. Okay, is called chemolithotrophy. Okay, for example, ammonia is converted into nitrate. Okay, and one of the example is of Nitrosomonas europaea. Okay, so there are variety of bacteria involved in this. Okay, when we will move forward, I will give you some more examples also. Okay, so most of the bacteria belonging to the metabolic type with carbon dioxide as their sole carbon or major source of carbon for their cell synthesis. They are also autotrophs. Okay, more specifically this group comprises of chemolithoautotrophs. Okay, chemolithoautotrophs, and all the chemolithotrophic bacteria, okay, so far examined, basically assimilate carbon dioxide by the fixation of the ribulose biphosphate cycle, which we studied in the photosynthesis in the especially dark reaction. Okay, the mechanism of this carbon dioxide fixation, okay, which is needed to be discussed in the case of especially carboxylobacteria. Okay, and some of them are even obligatory chemolithotrophic and some of them are even chemo organoheterotrophs also, or some of them are facultatively chemolithotrophic also, and many of the chemolithautotrophs are highly specialized and occupy a monopoly position. Okay, so the position, I think, of the ammonia, nitrite, or the inorganic sulfur compounds in nature is due to, in the first place, to the activities of nitrifying or the sulfide bacteria or the sulfur oxidizing bacteria. Okay, now moving further with the examples of chemoautotrophs. Okay, such as energy source in the case of ammonia, Okay, and the group will be nitrifying bacteria, and the genera will be Nitrosolubus, Nitrosomonas, and Nitrocystis. Next one, nitrogen dioxide, again nitrifying bacteria. Genera will be Nitrobacter, Nitrospina, and Nitrosococcus. And the nitrogen, these group do not belong to the chemoautotrophs, but simply for your example, okay, I have included, okay, nitrogen fixing bacteria such as Azotobacter, Anabenna, Prochlorococcus, and Rhizobium. Okay, other groups with examples sulfur bacteria such as Thiobacillus, Sulfolobus, Desulfatomaculum, Vollinella, Desulfovibrio, and Beggiatoa. Next one is iron bacteria, Gallionella, Sparotilus, Thiobacillus phaeroxidens. Okay, there is no comma here. Leptothix, Swanella, Onidiensis. Next one is a hydrogen bacteria, includes Ralstonia, formerly also known as alkaligenes, Nocardia, Xanthobacter, and Pseudomonas durxia. Okay. Next group of other bacteria also, methylotrophs, which includes Hyphomicrobium, Methylomonas, Methylobacterium, Methylocyanus, Paracoccus, Pseudomonas. Okay, this group use methane as energy source or alcohol as a energy source. So that's the reason their terms methyl, because use of methyl. Okay, now moving further with the methanogens, which is also a part of autotrophs. Okay, but we will not discuss in the case of chemoautotrophs. It will be discussed specially in a next point. Okay, so methanogens can use hydrogen, even carbon dioxide, formate, methylamine, trimethylamine, or acetate as their energy source. And the examples are Methanobacterium, Methanobrevibacter, Methanococcus, Methanomicrobium, Methanogenium, Methanospirillum, Thanosarcina. Okay, Methanospirillum, Thanosarcina. And the light ones, which are phototrophs, which we have already studied, Rhodobacter, Anabenna, again Prochlorococcus, okay, and Syniococcus, okay, and some of them are algae, which are not bacteria, okay. So now moving further with the chemoautotrophs, nitrifying bacteria, okay. So in the course of aerobic or anaerobic degradation of nitrogen containing substances, the nitrogen is liberated in the form of ammonium, and the formation of potassium nitrate during the composting of various animal manure is one of the most important and one of the old observations made by biologists. Okay, so this can be found in various other places also. Similar in the case of microbes. Okay, in the case of microbes, ammonium, which is liberated during the microbial degradation of the organic material, is diffused into the soil, where it gets oxidized by the atmospheric oxygen to yield nitrate. Okay, to yield nitrate. And this soil cover was basically, I think, the starting material for the production of potassium nitrate. Okay, so the conversion of ammonium to nitrite, that is nitrification, is basically carried out in soil by various nitrifying bacteria. Okay, and till now, as per current condition, we do not have any examples where a bacteria directly converts ammonia into nitrates. Okay, there are always two different kinds of bacteria involved in the reaction of conversion of ammonia into directly nitrate. Okay, of which one of them are ammonium oxidizers, as I told you earlier, and another group is a nitrite oxidizers. Okay, So some of the best known species are basically Nitrosomonas europea and Nitrobacter Vinogatsuki. Okay, Nitrosomonas europea and Nitrobacter Vinogatsuki, which were first studied by the scientist Sergei Vinogatsky. Okay, as he was the first scientist who succeeded in isolating pure cultures using strictly inorganic media. Okay, so this is the reason why Nitrobacter was named Nitrobacter Vinogradsky after the name of the scientist Sergei Vinogradsky. Okay, so according to the more recent uh, investigations, okay, species such as Nitrosolobus are very much involved, okay, as compared to Nitrosomonas. But Nitrosomonas are one of the main ammonia oxidizing organisms in the agricultural soils. But both genera are highly specialized for their oxidations. Okay, since the high concentration, I think high concentration of ammonium in alkaline soils have a toxic effect on Nitrobacter or Nitrosomonas and the Nitrosolobus. Okay, they also serve to improve environment for the nitrite oxidizers by both utilizing ammonium and increasing the acidity. Okay, increasing the acidity. Okay, but up to some extent. Okay, but up to some extent. So the nitrifying bacteria develop best under the neutral or alkaline conditions, since the oxidation of ammonia to nitrites result in the considerable acid formation, and the growth medium for Nitrosomonas must be well buffered, for example by addition of insoluble carbonates. Growth of both organisms is slowly minimal and general growth rates are very low. Okay, and as they are very slow growing bacteria. Okay, as they are very slow growing bacteria, and Vinogradsky also demonstrated that both the bacteria are obligate autotrophs and incapable of development in the absence of their specific inorganic nitrogen source. Okay, so the nitrifiers are basically gram negative bacteria, and they are collected in the family called Nitrobacteriaceae. Okay, in the family Nitrobacteriaceae, and the Nitrosomonas europea is an oval bacterium with a polar flagella. And in the case of marine habitats, okay, in the case of marine habitats, Nitrosococcus oceanus seems to be one of the organisms responsible for the oxidation of ammonia. And the nitrifying bacteria can be cultured in purely mineral soil solutions, but rather grow very slowly, and their generation times varies from about, you can imagine, vary from about 10 to about 20 years, 10 to about 20 years. As you all know, the generation time of E. coli, okay, generation time of E. coli is around 20 minutes. Okay, you can see the difference, how slow growing these bacteria are. Okay, as you all know, nitrifying bacteria are basically considered as strictly obligate chemolithotrophs. Strictly obligate chemolithotrophs. Okay, however, there are some of the observations which led that the Nitrobacter Vinogradsky was able to use acetate from the medium into the cell material. Okay, from the medium into the cell material. Okay, so now moving further with the oxidation of ammonia and the nitrite. Okay, the oxidation of ammonia is being carried out, okay, is being carried out by the Nitrosomonas europea or Nitrosococcus Arsenius or Nitrosospira branches or Nitrosolobus multiformis. So there are variety of bacteria, okay, and it is being done by the enzyme ammonia monooxygenase to convert ammonia to the intermediate hydroxyl amine. Okay, it will be converted into hydroxyl amine. Now next enzyme hydroxylamine oxidoreductase then catalyzes the production of nitrate, which is released into the environment. So okay, so this is the first reaction, okay, where ammonia will be converted into hydroxylamine and hydroxylamine will be converted into nitrite into nitrite, okay. And now the conversion of nitrite, okay, which is being done by the Nitrobacter Vinogatsuki or Nitrobacter agilis or Nitrobacter hamburgensis or Nitrococcus mobilis. Okay, so there are variety of bacteria, okay, but all of these are very slow growing bacteria, and this catalyze the conversion of nitrite to the nitrate, okay, with the help of enzyme nitrite oxidoreductase, with the help of enzyme nitrite oxidoreductase. And the reaction, the first step of the ammonia oxidation is basically an endergonic reaction, okay. And moving further, okay, there are also other nitrifiers, okay, there are also other nitrifiers which have some different type of mechanism, and they are included in the heterotrophic nitrification. So I'm not going to discuss about heterotrophic nitrification because we are right now discussing about the autotrophs, okay. This all are examples of a autotrophic nitrification, okay. So in the oil, sorry, not in the soil, the ammonium ions which are liberated, okay, are rapidly oxidized in the well aerated soils, okay, in the well aerated soils, so such as the soils which have very good amount of good amount of oxygen will be aerated faster, okay. Why? Because this reaction of ammonia oxidation needs oxygen, okay, it needs oxygen for the reaction to move forward. And the conversion of cation to an anion affects an acidification of the soil, hence leads to an increased solubility of the minerals such as potassium, calcium, magnesium, phosphates, etc. Okay, and the nitrifying microflora are therefore regarded as significant factor in the soil fertility. Significant factor in the soil fertility, and ammonium ions are basically retained in the soil, okay, more retained as than the nitrite because they are involved in the formation of humus also, okay. They are also involved in the formation of the humus also. You already have studied humus in your semester 4 of microbiology unit 1, okay. And nitrite on the other hand can be easily leached out, okay, because it is being characterized by various denitrifying bacteria where it will be converted into nitrogen again, okay, it will be converted into nitrogen again, okay. Now the metabolic pH which will be needed to carry out this reaction is around 7 to 8, is around 7 to 8 where this reaction takes place, okay. And nitrifying bacteria are also indirectly involved in the destruction of limestone cements because they oxidized any ammonium derived from the atmosphere or animal excreta to the nitric acid. So these are some of the other detrimental effects of the nitrifying bacteria also. Okay, and one of the most important ecological importance of the nitrifying bacteria, in which especially ammonia oxidizers, is basically in the ammonium salt fertilizers. Okay, under the conditions of the oxygen limitations, okay, it have been shown that the nitrosomonas and nitroso can carry out nitrite respiration. Okay, but under the waterlogged conditions where there is very less amount of oxygen, the ammonia oxidizers can be a boon for a farmers. Okay, so as I told you, some of the denitrifiers can also decrease the fertility of the soil also. Okay, now moving further. Okay, so the mechanism, okay, is shown here. Okay, ammonia oxidizing bacteria, it will convert ammonia, okay, it will convert ammonia into nitrites, and this is another one. Okay, this one is an archaea, and this one is an ammonia oxidizing bacteria. Okay, this is ammonia oxidizing bacteria, and this will be passed on to the nitrite oxidizing bacteria. This diagram is just for the representation purpose. Okay, if you want to write in the exam, you can use this reaction, and this description is given. This description is given here. Okay, in the exam you need to write this reaction. Okay, the diagram, the photographs given here is just for the representation purposes only. Okay, now moving further, the oxidation of ammonia to nitrite is carried out by Nitrosomonas, Nitrosospira, Nitrosovibrio, Nitrosococcus species, and they are all obligate chemolithotropes, and also nitrooxidizers, Nitrobacter, Nitrospina, Nitrococcus, with the exception of Nitrobacter strains. Because the nitrite is very toxic to most of the organisms, the process of nitrite production and nitrite oxidation are one of the remarkable ones. Okay, now the obligate phototrophy, okay, not phototrophy, autotrophy, and which regarding to the types of bacteria and its speciality. First one is Nitrosomonas. They are rod shaped, flagella subpolar, and intracytoplasmic membranes are basically laminars. Okay, GC content is for their identification. Okay, and they are obligate autotrophs. Almost all of them are obligate autotrophs except Nitrobacter, which are not obligate autotrophs. Okay, of which Nitrosomonas, Nitrosospira, Nitrosococcus are basically ammonia oxidizers. Nitrobacter, Nitrospina, and Nitrococcus are nitrite oxidizers. Okay, and the shape of Nitrosospira is a type spiral in shape, and the flagella peritrichous. Nitrosococcus similar to round shape, flagella are peritrichous. Okay, and the shape of Nitrosolobus is they are lobed. Okay, what do you mean by lobe structures? It may have some type of lobed, okay, fixed structures which can be used for their identification. Okay, now the Nitrobacter, they are rod or pear shaped. Nitrosospina, they are long or a very slender rod. Nitrococcus, they are spear in shape. They are spear in shape, that is round one. Okay, now where are these bacteria found? Okay, where are this bacteria found? Ammonia oxidizers, in which first one, Nitrosomonas europaea, is found in soil, sewage, marine, okay, or even fresh water. Nitrosococcus orsini is found in marine environments only. Okay, it's found in marine environments only. Next one is Nitrosospira branensis. It is found in the soil. Next one is the nitrite oxidizing bacteria, of which the first one is Nitrobacter Vinogatsuki, which is found in soil, freshwater and marine environment. Next one is Nitrococcus mobilis. It is found in the again marine environment, but these are not obligate marine ones. Okay. Now these are some of the photographs. These are some of the photographs of the various bacteria. Okay, nitrifying bacteria. This one is a phase contrast of Nitrobacter Vinogatsuki. Okay, Nitrobacter Vinogatsuki. Again, with the cap of cytomembranes, you can see here. Okay. Okay, cytoplasmic membranes, you can see clearly here, extensive ones. Okay, now this one is a lobe structure. Okay. Now moving further with the chemoautotrophs. Okay, Vinogradsky, in addition to the nitrifying bacteria, he also studied sulfur oxidizers. Okay, he also studied sulfur oxidizers. Vinogradsky examined the properties of filamentous gliding bacteria of such as Begoscia, Thiothrix group, which occur characteristically in the certain sulfide-rich environments and often contain a massive inclusions of the elemental sulfur. Okay, this sulfur can be easily be identified using a specialized microscopy. Okay, he also showed that these organisms can oxidize hydrogen sulfide initially to the elemental sulfur which accumulates in the cell. Okay, and this sulfur can be easily be identified in their cell structures also. Okay, so a wide variety of other aerobic bacteria have been subsequently shown to oxidize hydrogen sulfide also in a similar manner with the transient intracellular sulfur deposition. Okay, and their ability to obtain energy by the oxidation of reduced sulfur compound is the property of a group of gram negative bacteria polar with the polar bacteria. Okay, sorry, polar flagella, and one of the main genus is the Thiobacillus. Okay, there are also other ones also such as spiral shaped, which is a Thiomicrospira, and one of them is thermophilic also, which is basically a sulfolobus. Okay, so the bacteria which oxidize hydrogen sulfide with the formation of intracellular sulfur deposits are, the first group is the filamentous gliding organisms, which includes Begoscia, Thiothrix, Thioploca. Next one is a very large unicellular gliding organisms, of which the first one is Achromatium. Okay. Next one is large unicellular rod-shaped or spiral organisms, immotile or motile by their flagella, of which the first one is cell rod-shaped immotile Thiobacterium, cell rod-shaped polar flagella Macromonas, cell round or ovoid Peritrichus flagella, Thiovulvum, cells spiral polar flagella Thiospira. Okay, and each of the these bacteria have some different pH for their growth. Okay, so the most of the common sulfur bacteria use different compounds, okay, and mostly as we are going to study just oxidizing, okay. In this case, electron donors are hydrogen sulfide, elemental sulfur, or even thiosulfate, or even sulfite, okay. In most cases the final oxidation product will be sulfate, okay, final oxidation will be sulfate, okay. And sulfide oxidation occurs in the stages with the first oxidation step yielding elemental sulfur, okay. And some sulfur oxidizing bacteria such as Begoscia deposit their any elemental sulfur inside the cells, where sulfur exists as a potential energy in the form of electron reserve, okay. So when the supply of the sulfide has been depleted, additional energy can be conserved from the oxidation of sulfur to the sulfate. So when the elemental sulfur is present externally, the organism must attach itself to the sulfur particle, because elemental sulfur is basically an insoluble compound, insoluble ones, okay. So here is a photograph of a Begoscia, okay. You can clearly see here white dotted ones are basically internal sulfur granules, okay. Now given in the diagram B, okay, which is attachment of cell of the sulfur oxidizing archaean sulfolobus acidocaldarius, okay, to a crystal of elemental sulfur. You can see here clearly these are the cells, okay, and these are the crystals of the sulfur. These are the crystals of the sulfur, okay. In order to visualize these cells, they were visualized in the fluorescence microscope with the help of dye acridine orange, okay. And this black coloration is due to the sulfur, because sulfur crystal will not be able to stain, okay. So they will not show any of the fluorescence, okay. So there will be some type of dark structures, whereas bacteria, you can clearly see here, okay, they will emit fluorescence through which we can identify the bacteria, okay. So now with the sulfur oxidizing bacteria, okay, if you want to write down, please write down. I'm giving some other examples also. uh, One of them is thiobacillus thioxidans, and the pH requirement for their growth, okay, is around 2 to 5, okay, is around 2 to 5, okay. And the electron donors which they use is elemental sulfur sulfides, okay. And they are basically obligate chemoautotrophs, okay. They are obligate chemoautotrophs, okay, or more specifically they are obligatory autotrophic. The second example is of thiobacillus ferrooxidans. The second example is of thiobacillus ferrooxidans, which have a pH of about 2 to 6, okay, which have a pH of about 2 to 6, okay. These use sulfides and elemental sulfur. These use sulfides and elemental sulfur, and they are facultatively autotrophic, and the third one is a thiobacillus thioparus, and the pH of their growth is from 6 to 8, okay, it's around 6 to 8. And there you use again sulfide and sulfates, sorry, not sulfates, elemental sulfur and they are obligatory autotrophic, okay. Now one example of archaea which we studied, which we discussed one slide before, is a sulfolobus, acidocaldarius, sulfolobus acidocaldarius, and its requirement for growth of the pH is 2 to 3, is 2 to 3, and they use elemental sulfur, for electron donor, glutamate also, they even use peptone also, and they are facultatively autotrophic, okay. And some other ones such as sulfolobus, besides sulfolobus acidocaldarius, Cadrella acidophila belong to the Archaeobacteria and are found in the extreme ecosystems also, okay. So they inhabit acid hot springs of various volcanic hydrogen sulfide is, where hydrogen sulfide is oxidized, okay. So how they convert, okay, sulfurs into sulfates, okay. So in this case as shown in the diagram, here sulfides, okay, are converted into polysulfide before being oxidized further, okay. They are converted into polysulfides, okay, you can see clearly here, okay. And the thiosulfate is metabolized to sulfide and polysulfide by rhodanase, by the enzyme rhodanase, you can see here, okay. Polysulfates, thiosulfates, okay, or directly oxidized to the sulfate by the thiosulfate oxidizing multi-enzyme complex, okay. And the sulfur compound oxidation is coupled to the reduction of cytochrome B or C, okay, is coupled to the reduction of cytochrome B or C. So starting with the process, well, the sulfur, okay, will be converted into elemental ones with the help of enzyme sulfide oxidizing enzyme. Now the second reaction will be conversion of sulf elemental sulfur, okay, with the help of conversion to polysulfide also, okay, which is results to conversion of polysulfide, okay. And the third enzyme which will be involved will be rhodanase, okay, will be rhodanase, and this polysulfide will be directly converted into sulfate, okay, with the help of the thiosulfate oxidizing multi-enzyme complex, and this sulfur will be converted into SO3 2 minus, okay, with the help of sulfur oxidizing enzyme again, okay, okay. Now moving further, this will be converted into APS, okay, APS with the help of enzyme APS reductase, okay. APS is adeno poly. What? How many of you know APS? How many of you know APS? What is APS? How many of you know? Okay, we have already studied in some cases also. No problem. This APS will be further converted into sulfates with the help of enzyme ADP sulfurylase. Okay, ADP sulfurylase. Okay, which is basically a adenyl transferase, and this SO3 minus will be directly converted into sulfate with the help of enzyme sulfide cytochrome C reductase, Okay, so it has been difficult to establish these steps of the pathway, and there are variety of protein complexes also involved in these reactions also. Okay, and it is assumed that the electrons obtained in the oxidation of sulfide to sulfate enter the respiratory chain at the level of cytochrome C, and at least some thiobacilli such as Thiobacillus thioparus or Thiobacillus denitrificans can utilize energy available from the oxidation of sulfide to the sulfate by substrate level phosphorylation also. Okay, so it depends on various other forms also. So in the case of Begosia, which is found in the sediments of ponds, okay, or slowly moving waters, okay, Sergei Vinogatsuki used Begosia to carry out these investigations and led to the concept of chemoautotropy. Okay, and this Begosia failed to grow in a pure culture in the laboratory, and with Begosia can be studied with the help of the Vinogradsky column. Okay, does anyone remember APS now? Still no answer? Okay, I will give you a hint. It is related to the adenyl. Okay, it is related something to the adenyl. Okay, now photographs of the sulfur oxidizers. Okay, a macro photograph of cells of thiovulvum, yellow dots as seen here. You can see the yellow dots, okay, that form a thin wheel around the marine sediments. You can see here some of the wheels one, okay, containing a hydrogen sulfide, large irregular structures. You can see the irregular structures, okay, such as sand grains. The thiovulvum wheel is employed as a strategy for regulating nutrient flow, in particular obtaining hydrogen sulfide and oxygen for the energy needs. Now you can clearly see the sulfur globules in the dividing cell of a thiovulvum. You can see clearly here. These are the sulfur globules which are present in the various sulfur oxidizers also. Okay, if you want to see more photographs, you can simply search sulfur oxidizers photographs. Okay, you will get variety of information. Okay, now a small video. Okay, now a small video. A bacteria from a sulfur spring. It's a video by I think downloaded from YouTube. Okay, but the author is Bill Kelly. cells assimilate carbon. They are also present in the activated sludge. Okay. And iode takes present gliding like motion similar to that of found in the cyanobacteria. Carbon dioxide. Purple color, as we already discussed, is the light absorbing. The light absorbing pigments. With the help of phase contrast microscopy we can identify, but in the bright field microscopy you can see the difference. Okay, this is video by Bernard.